dear Eugen, for this kind invitation to be here. I'm honored to present the talk for this.
Here, always we use it.
For this 20th year anniversary. It's very nice to be here and to see the progress, because I don't remember exactly the day and the year when I was here first, but the progress is visible. So congratulations, and I'm happy to present you a talk on something which is attractive, magnetic, yes. So I will use this. The outline of my presentation is presented here. After a short introduction, I will present you some procedures, synthesis of magnetic nanoparticles, magnetic clusters, ferrofluids and bioferrofluids. I will try to make the difference between ferrofluids and bioferrofluids. So, first, the conventional classical ferrofluids. You can see here the main characteristics these should have. So first, the particles, which are usually 99% iron oxide. Of course, there are iron also, cobalt also, but all what is in use actually is iron oxide. Iron oxide, which may be magnetite or megamite, but iron oxide. Okay. The particles should be really very small to have what we mean ferrofluid. What is a ferrofluid actually? Here I try to outline this. Colloidal suspension in a carrier liquid, okay, from water to a lot of other liquids we will see immediately. What is the reason for these such small particles? We have to manage to have a colloid which is in this conventional ferrofluids indefinitely stable. This cannot be achieved otherwise than going to be smaller, when exactly the sizes than 10 nanometers. This 10 comes out from here. That is the magnetic interaction versus the thermal movement of particles. And this dictates you should be less than 10 nanometers. Of course, if you have this material, this works like a Langemann type magnetic material. It's all the characteristics which you are used to be for a magnetic material, except one essential. It is liquid. So the magnetic nanoparticles confer what we can define as magnetizable liquid, which is we cannot find in the nature. So we have to make it. That is the problem. So in the ideal case, we can suppose that this is a magnetizable continuum. But we will see that this continuum has something which is not so easy and not so simple. Stable. What it means, colloidally stable, indefinitely stable. We have here is a sample in our lab which has already 40 years. The magnetic colloid, the ferrofluid, was pushed on a big magnet, 1 liter approximately, the ferrofluid, enough strongly magnetized, and is on the magnet for 40 years already. And doesn't show any behavior which would give you the signal that is the sedimentation there. So it's a perfect magnetizable continuum. Okay. What makes this possible? The stabilization. This is a mechanism we apply usually the spherical mechanism for stabilization. That is, each particle is coated with an organic layer. The most frequently used are the carboxylic acids. And the most frequently used carboxylic acid is the oleic acid, to be very clear. Well, depends on the carrier medium, that is, for example, non-polar organic carriers. We need only one layer of oleic acid to be more precise, but it could be okay, lauric acid, malic acid, and so on. And in water, to give a complete example, a polar, very polar liquid, we need two layers in order to ensure the hydrophilic outer region. In these conditions, we have a really good, as I have shown you, good ferrophilic. Now, which is important for what I would like to say further, is the various sizes of these particles. Usually, a nanoparticle of this range, this size range, about 5 to 10 nanometers, has a problem on the surface. The so-called spin disorder, which makes the outer layer non-magnetic, doesn't contribute to the magnetic moment. And this is really an issue for the overall magnetization. And this layer is about 1 nanometer. Okay, so we have the so-called physical size, which is, we can see on the transmission electron microscopy, yes, and the magnetic side, which is smaller. You can see it here. And the third one is the outer, together with the outer layer, the so-called hydrodynamic diameter, because of course the surfactant layer moves together with the particles, of course. So these three dimension sizes are very important in the behavior of a magnetic fluid, ferrofluid. For many applications, including these nanomedical applications too, the different sizes in between the 5 to 10 nanometer range dictates also something, an interesting behavior in what concerns the magnetization, relaxation. So if we have enough small particles, then the movement, the rotation of the magnetic moment can be done by spin rearrangement, so the particle itself doesn't move in the liquid medium. If it's a little bit larger, somewhere 9-10 nanometers, it's already a so-called rigid dipole. That means that the magnetic moment is frozen in the particle, so if you have to move the magnetic moment in the direction of a field, you have to move all the particle. So, in the first case, you don't need any movement in the liquid, but in the second one, the hydrodynamic diameter dictates this movement in the viscous medium. This is very important, for example, in an application which is in nanomedicine now, it's very frequently you can see in papers, is the magnetic hyperthermia. There is both relaxations are involved and so on. Okay, let's go then to this synthetic image. What sizes are involved actually in what we call ferrofluids? And the conventional one is here, conventional or ideal, I give this name, between 5 and 10 nanometers, the sizes. These are the solid physical sizes. In real ferrofluids, which are used all over the world in engineering applications, this is a little bit larger, from 5 to 15 nanometers. And what we call bioferrofluids, this is really is the outcome of the last 2 decades, and it's a very different picture. So, if we look to contrast media, we have sizes between 3 to 5 nanometers. This is a big price for the magnetic moment, but seems to be okay for this type of applications. Also, we have particles much larger than 15 nanometers. But in order to keep the superparamagnetic behavior of the wall, we cannot go with physical sizes to these large sizes, of course not. So we make clusters, look. And these clusters are much larger than 15 nanometers, and overall they have the superparamagnetic behavior, which is essential for any application. But we exceed much the 10 nanometers which is needed for colloidal stability. So do not expect that these bioferrofluids, except the left-hand side, which are perfectly stable, to remain stable for a long period of time in magnetic field. No, it's out of case. But their use and usefulness is defined on their behavior in the biological media. What procedures we apply? I would like to say that we have our lab in Timișoara. Dr. Turcu, Rodica have laboratories in Cluj, so this is an overall picture of what we are doing in synthesis of particles, nanoparticles. By chemical co-precipitation, here probably most of you know that this is the most popular, perhaps the most simple, and so on, and the most cheap procedure. Also thermal decomposition, which is a little bit more precise, gives you particles, very nice particles, sometimes very tricky. I cannot lose the time for now to explain to why saying this. But okay, these are very interesting. Both procedures are used overall, and the best one are these two in manufacturing magnetic nanoparticles. Okay, clusters, because we have seen that most of today's bioferrofluids need clusters. The clusters can be prepared by mini emulsion process using ferrofluids, that is single particles in a mini emulsion process to make clusters. You can make them directly in one-step procedure, solvothermal, or in a fast precipitation procedure I will show you a little bit later. Now, the last part is to disperse this, and in our field of activity now is water, essentially. It is aqueous ferrofluids are the most used in biotechnology or nanomedicine. So, this is the most used procedure. I told you that is chemical co-precipitation. We upscale this procedure, I will show you. Also, the mini emulsion for cluster preparation, magnetic nanocluster preparation is a very well-defined procedure. We proceed this in many years, use this in many years along, and now it is upscaled also. And here is the solvothermal procedure, also upscaled recently. Now, this is the core of, to say, of my presentation. That is the manifold characterization. You cannot use a system of magnetic nanoparticles, any nanosystem, without a very careful characterization. Starting with microscopy and various other procedures you can see here, XPS and magneto-optics, ELS, of course, and more advanced structural examination processes like small angle scattering with X-rays or neutrons also can be used in some cases. And of course, magnetics, which is very important, flow properties, magnetorology, and if you have applications which require, you have to see the radiation resistance of the ferrofluids. Here you have absolutely usual real, and I say engineering ferrofluid. I mean the nanoparticle sizes which are in between the limits I told you, and this is produced already at our industrial company get this technology many years ago, and the result is very good, and they can deliver such ferrofluids each day. Here, the particles produced at the Institute of Rodica Turcu, in ECDT, in Romania, everybody knows it. It's very interesting to see that not only the spherical particles are interested, and if you think about magnetic hyperthermia, you can think about nanotubes and so on. So this is already what we are doing now, and it looks to future development. Also, you can see the sizes, the different sizes, depending on the application we are foreseen for this type of particles. Here, the images of the nanoclusters prepared, magnetic nanoclusters, by mini emulsion procedure. Densely packed nanoparticles, magnetic nanoparticles, which give a very good behavior, magnetic behavior, that is paramagnetism is ensured, and the sizes are regulated by the process, which is well known at this time. Also further, with various functionalizations specific for biotechnology and nanomedicine, hyaluronic acid coating, for other, for example, polyethylene glycol coating, which are very good for such kind of applications. Okay, and here the solvothermal synthesis. What is interesting for solvothermal in particular? You do not have individual particle coating, only outer coating. So the particles are even more closely packed than for the mini emulsion process. This is to be mentioned. Now, to give some examples of characterization for engineering ferrofluids first. Here you have a magnetization, a full magnetization curve here, and also here is the low field region, which shows you that no difference between various concentrations. If you look here to the magnetization, from very small magnetization up to the highest possible. Actually, ferrofluids with magnetite can attain 100 kilaampere per meter, that is approximately 1,400 Gauss, but these are already a kind of not very good flowing material, but up to 1,200 Gauss we prepared in the lab. It's also prepared by our industrial company, so this is a good limit. And the hydrodynamic volume fraction, you can see here, is very close to the high package 0.75, which is, well, only for the identical spherical particles can be extended to 0.8, but the flowing characteristics are good up to this magnetization. And what we can see from here is that the interactions are well screened. This is very important by this stabilization layer. Here, the rheological behavior, the volume fraction dependent viscosity is a very important characteristic of any ferrofluid. And here is also the same conclusion it's a very interesting model here for non-magnetic colloidal suspensions. The interaction parameter in theory says this value, and we fit it out for our magnetic particles, which interact, of course, and it's very close to the non-magnetic particles, which means, again, the conclusion that if you stabilize well the ferrofluid, you can screen the attractive interactions which destroy practically the ferrofluid. Okay. So, a little bit more fine analysis of the interactions is due to busbar spectroscopy, which was done by our colleague Kunce Victor in Bucharest. And what shows? I selected two samples, a very diluted one and a very concentrated one. The concentration makes, of course, the interactions, the dipolar interactions increase, which were not seen in rheology, were not seen in the magnetization curve, but it evidenced very well in reservoir spectroscopy. And here you have this dependence of the anisotropy energy depending on the packing. It shows up that the interactions exist and they can show up, but they do not cause clusters, which is very important for a good therapy. Now go to scattering methods. We practiced this about 25 years like that. And here you have a difference between a very stable ferrofluid here, the scattering pattern, and the less well stabilized, the water-based ferrofluids. Here I would like to mention that water is the most difficult carrier possible. So if you look in the literature overall on ferrofluids, you will see that the overall issue is the formation of clusters, the stability which is not so long term as we would like to have, and so on. The main reason is the high polarity of the carrier, of course. So the stabilization has several issues to solve. Well, here is the scattering pattern. We show that we have aggregates, fractal type aggregates, and here we do not have any such, up to highest concentration. But this is only an example. Let's see this overall characterization, which we did more recently, on two very different aqueous ferrofluids. One, which is use electrostatic stabilization. We do not practice this. These are colleagues from Finland who practice this. Actually, Professor Massa from Paris invented this in the early 80s. And our ferrofluid, which originates from Shimoizaka from Japan, the so-called double layer stabilization mechanism, I already show up for you. So these two very different and competing procedures, now we have to compare. Up to the highest concentration achieved until today, or 2 years ago, or 4 years ago, excuse me. Actually, in publications, these are the most concentrated samples prepared anyhow until today. So the electrostatic stabilization achieved about 1,000 gauss, which is very high water. You can get it. Excuse me. You can look here. Yes. At the same time, the other one, this double layer spherical stabilization, is only 600 gauss. The reason is very simple. You have the hydrodynamic volume is the same, 30%, but the outer layer in the steric stabilization is much thicker, 5 minutes. So the difference between these two ferrofluids resides in the preparation mechanism, and you can see the values here, about 6 nanometers for the citric acid stabilizer and about 7.8 for the double layer oleic acid sample. Now scattering, to see how it looks, the two kind of scattering methods we used. First, the small angle X-ray scattering, which was done at DAISY, the Dutch Electron and Synchrotron, the Biosax facility, and the other one was the small angle Newton scattering, was done in the Physical Institute of Energy in Kjeller in Norway. Here, excuse me. Here is their small angle Newton scattering facility, and the result. The electrostatic stabilization don't show up clusters. Look, the structure of the scattering curve. And the double layer oleic acid samples show up clearly the structural formation. So these two techniques are very strong in characterizing up to the highest concentration of this kind of therapies. Rheology, magnetorheology show up the same thing. So practically no viscosity change in magnetic field. And due to the clusters, the oleic acid stabilized samples show increase of viscosity. So the conclusion is that mostly in the electrostatic stabilized ferrofluid we have most individual particles, but in the other one, the spherical stabilization, we have also clusters. Now, some applications shortly. All of the applications which fit to our meeting used our nanomaterial, it is magnetic nanoparticles from clusters, and colleagues from abroad, usually abroad, use them in various processes. One is this handling of antibodies, actually separating antibodies. You can see here the process relies on splitting of droplets of ferrofluid. That is, you can increase the surface of the ferrofluid exponentially by a magnetic field. Please look here. So you have a large surface, functionalize the surface, and attract the molecules you need. That is, very shortly speaking, the procedure. Another one, use also a ferrofluid, water-based ferrofluid, for histamine detection in red wine. I think it's a good idea for today, maybe even for tomorrow, how to detect histamine in a good red wine, which we suppose it is good. It uses a functionalization of the double layer, oleic acid double layer coated magnetite nanoparticles, and the detection is based on the fact of the hydrodynamic volume and the magnetic signal they produce. They don't need applied magnetic field. It's an AC susceptibility measurement actually, and this gives the result. Of course, it needs a lot of things to put together this. Here the clusters are used for other biomolecules. It's a similar procedure. Here a bioseparation method which was done on micropilot scale already in Germany at KIT, Karlsruhe Institute of Technology, by our colleagues. And look here the comparison between the particles prepared, the clusters actually, prepared in Cluj, compared to various commercial samples. So comparison is relevant, it is a very good material which we produced in this cooperation. Now, here is a much larger particle, 1 micron, 2 microns, 3 micron range, prepared at Yashi, in the Institute of Micromolecular Chemistry. And what is very interesting, and I try to show you up that the Sapar paramagnetism is conserved because we use aqueous ferrofluid, and the magnetic nanoparticles decorate the large calcium carbonate microspheres. What is interesting here, the magnetic moment. Apparently the magnetization is not so big, 3 mg per gram is not so big, but taking into account the volume of the particles is about 3 orders of magnitude larger than the usual clusters. So magnetic separation, very nice. Here is a essentially nanomedical application which looks at this at the beginning only still. That is the magnetic stents, the use of magnetic stents in cardiovascular diseases. Here is the stent. Here is the flow analysis in such a stent, which is very complex. And we use the suspension of polyethylene glycol coated magnetite clusters. And they are captured by this ferromagnetic stent. And the idea is that this nanocluster is coated by the necessary medication for after the surgery, the patient having this kind of diseases. Okay. Here, this is already an application in environmental remediation for uranium removal. Our colleagues from Cyprus, for the long cooperation with them, prepared a magnetoresponsive membrane functionalized, which the adhesion of adhesive forces with uranium is advanced. So the separation is, you can see the removal rate and the desorption rate from these composites. And this is a promising application too. And here is already industrial application in Chernobyl, nuclear power plant. We use magnetic seals from 2012, so now we have 10 years already, to practically reduce to zero the tritium escape. And here is the precision. They verify this periodically, therefore I cannot say zero, because you have this value. But practically, the escape now is zero, the tritium escape. So this application shows that magnetic seals are really useful even in this field of application. So, 2 slides still. One is from Cluj, that is these magnetic nanoclusters are produced on large scale already at our industrial partner, Romanian Steel Corporation from Odorheiu Secuiesc. So you can use them. Quantities are no problem. And a lot of type of ferrofluids which are produced in our lab, but some of, and a lot of them are produced by the Romanian Steel Corporation. So there are available individual, singular magnetic nanoparticles on relatively large scale. A lab scale procedure gives you 60 grams. So the industrial one gives you even much more. So this is the mixed team, Cluj, Odorheiu, Timișoara. They made this technology transfer. Here are the partners. We are working in Timișoara, Cluj, Bucharest, and various other European centers of research. We develop partnership along the time. So this is a characteristic picture. Thank you.
Okay, no time for questions, I'm sorry. During the coffee break, fine?
Okay, thank you.
So, next presentation comes from the Bologna Institute of Molecular Chemistry in Iași, Institute of Academics. A group of researchers, Alex Romano will present our results. And Alex traveled in many laboratories. A few years ago he came in Iași, and we decided to stay in Iași. We are very happy with him. I hope he is very happy with us also. Alex, please.
I confirm.
Thank you very much, Professor Smenes, for this kind introduction. Also, I would like to thank you again for inviting me to this symposium, because for us as chemists, it's very important to share the results with the community of interdisciplinary researchers and to get a feedback. And in the current presentation, we actually developed two completely different methods to the production of probes for both the fluorescence imaging and for the Raman imaging using supramolecular chemistry. And I would like to start the presentation. It's not working. Thank you. With this beautiful picture, and actually today morning we had amazing talks referring to the fluorescence imaging, and it will prove that this technique actually is still great, together with other techniques, of course. And this picture also confirms that because already 20 years ago it was possible to simultaneously image 5 different cell components with 5 different colors. This is amazing. Well, nowadays we have a lot of fluorescent probes which are commercially available and which are easily used for the obtaining of great pictures. And among these, I would like to mention the samples, fluorescent samples probes who are sensible to the pH changes. Their fluorescence is extremely dependent, and they are very important because they could actually visualize some of the processes within the cells which are accompanied by the change in the pH. And I outlined only a few of the samples because I was interested personally in compounds which do change their fluorescence by the protonation of the nitrogen in their structure. That is why, because a couple of years ago, we were employed in a project with our colleagues from the recycling chemistry department, and they were investigating this highly fluorescent compound, which is an indolizine pyridinium salt. It is easily obtained by a 3-step synthesis. I will not go into deep in it, but this highly fluorescent compound has a indolizine moiety within it, which offers it a huge fluorescence, and also it has this quaternary pyridinium salt, which is actually offers the pH sensibility towards the fluorescence. More precisely, this nitrogen, quaternary nitrogen, actually in the basic media loses a molecule of hydrobromic acid and is transformed in a very low fluorescence zwitterionic indolizine, and this process is reversed. The problem with this compound, as with many other organic fluorophores, is that it has 2 main disadvantages which very much limited their biomedical application. And more precisely, these compounds are not soluble in water, they have very limited solubility in water, and secondly, they are toxic. Well, we decided to apply some supramolecular approaches to solve these 2 problems. And I'm referring to the formation of supramolecular complexes, which are called host-guest inclusion complexes, with the cycloidic strings. These cycloidic strings are cyclic oligosaccharides. They have a very particular structure because of their molecular components which are hydrophobic are oriented toward inside of the molecule, while hydrophilic parts are going outside the molecule. Well, it creates an internal cavity which is hydrophobic, and if a molecule is fitted in that cavity, actually it can go inside, and due to the hydrophilic environment, the compound gets solubilized. Well, in our case, we use the beta-cyclic stream, and we perform the reaction with the cyclic stream and the compound we were interested in, and we observed that in the reaction where the ratio was 1 to 1.5 of cyclic stream, we could obtain complete solubilization in water, which was very great. It means that probably a kind of inclusion complex like that is formed. What we did immediately is to check either this complexation is affecting the fluorescent properties of the initial compound, so we compared the compound and the inclusion complexes. So, well, as I mentioned, the compound is not very fluorescent in the basic media, then at physiological pH it gets more fluorescent, and then it's highest fluorescent in the acidic media. While the inclusion complexes, or supposed inclusion complexes, showed similar behavior at the basic pH, but then at physiological pH and acidic pH, the fluorescence was actually similar. And that was one of the indications that probably the complexation took place because the transition from the salt to elite and back is actually accompanied with some transformation of the structural changes. And if the complex is formed, then these transformations are very hindered. It means that the rotation of the molecules is hindered, and then probably the fluorescence emission is also affected. Well, the easiest way to figure out if the complex is formed or not is to use the mass spectrometry, and particularly the electron spray ionization mass spectrometry. It immediately gives you information about the formation or not of the complex. And we could see in the spectrum that we find the mass which corresponds exactly to the compound which forms the complex with cyclic in the ratio 1 to 1. But then we investigated further the spectra and observed that this compound can not only form the complex of 1 to 1, but also can form 1 to 2. Here is the mass of it. Well, it was a little bit not really weird because it's very often happening, but still we decided to apply for theoretical chemistry and to explain this process. We did some simple theoretical calculations, and more precisely we did molecular docking on the optimized molecules. And we observed that in case of one-to-one, indeed, the molecular docking showed us that it is possible to form the complexes. It looks something like this cartoon, where the cyclodextrin covers the middle part of the molecule, meaning the salt and the indocyanine part. The calculated theoretically binding energy and dissociation constant actually prove that, well, it might be possible the formation of these complexes. Then we investigated the possibility of two cyclodextrin molecules to form an inclusion complexes with one molecule of the dye. And again, the molecular docking studies showed that two molecules can participate in the formation of the complex. Then in this case, the cyclodextrins will be positioned at the marginal phenyl groups. And surprisingly, the binding energy and the dissociation constants were much better, explaining that the possibility of the 1-to-2 complex formation is much more preferable. Well, experimentally, we tried, it was very good for us, we wanted to shift completely the reaction towards the formation of only one species, and then we said, okay, we increased the amount of cyclodextrin in the system, but still, we had a mixture of 1-to-1 and 1-to-2 experimentally. Nevertheless, we had a transparent solution, so we decided to do the first test if the cyclodextrin is indeed inhibiting the toxicity of the compound. And we tested the cell viability assays on two cell lines, on the HeLa cell and fibroblast, and here was the first extremely important result. Well, the compound showed in green, indeed, at any given investigated concentration, killed everything. It was just completely killer compound. But surprisingly, the inclusion complex solution we investigated was extremely viable. It was really a shock for us. We didn't expect such a good result, but apparently it's happening. We repeated 3 times the experiment, and every time it was giving great cell viability test results. Well, in case of fibroblast, the tendency is almost the same, but since the fibroblast having a very slow, let's say slower metabolism comparing to the HeLa cells, well, the results are slightly different, but mostly the same. The compound is killing almost everything, well, less concentration, but then the inclusion complex is less toxic. Well, then evidently the next step was to try and see if these inclusion complexes do penetrate the cell membranes, and we can use them as a markers for cells. Again, these two cell lines were treated, and I hope you can see, were treated with inclusion complexes, and after 15 minutes we observed that yes, indeed, the compound did penetrate inside the cell, it was accumulated in the cytoplasm, but the very great effect came after 24 hours of incubation. The same cells showed a completely different picture the next day. They were much brighter, and besides the brightness, we could observe that in both cell lines we could see specific accumulation within the cytoplasm. Well, in case of fibroblasts, this accumulation was not so evident, but in case of HeLa cells, it was amazing. So what we thought, that taking into account the nature of the compound, that it prefers to increase the fluorescence in acidic environment, we thought that these compounds may actually accumulate in lysosomes. And to prove this statement, we decided to do a colocalization experiment using the commercially available lysotracker red kit. Well, after 24 hours of incubation, and actually this fluorescence stayed for 48 hours and also 72 hours, we then treated the same cells with the lysotracker red and performed the overlapping. The software showed extremely nice overlapping coefficients, so showing that indeed we were right, probably and actually proven that our inclusion complexes do prefer accumulating in the lysosomes. Sounds great. Then we tested another 3 compounds, labeled A, B, and C. Well, as chemists, we were interested in influence of the substituents at the marginal part on the stability of the complexes. Nothing interesting. They behave exactly the same way as the previously explained compound. Well, the only difference was that in transmission electron microscopy we saw different mechanisms of aggregation. That's nothing to do with the topic of this symposium. But what we did additionally, we thought that, okay, we saw that the compounds are accumulated in the lysosomes, they are acidic ones, but what if we will check if it can accumulate also in neutral acidic or in neutral organelles of cells? And we decided to check if the compound is present also in mitochondria. And for that purpose, we also prepared the cells incubated for 24 hours with inclusion complexes, then treated it with a commercial MetaTracker red, and overlaid the images. And the software said that yes, there is a good overlapping coefficient, but it's not so good like in case of lysosomes. So we draw a general conclusion that maybe it's more preferable to stay in lysosomes rather than mitochondria. Well, we like this project very much. The compound, which even we didn't know if the inclusion complex is 1-to-1 or 1-to-2, we adopted this protocol in our group for staying in cells independently of what kind of project we do. For example, we recently published an article where we prepared supramolecular hydrogels which were enforced with carbon nanotubes. And being carbon nanotubes inside the hydrogel, they were completely dark, so we needed to dye it to color, and of course we came to our inclusion complexes, so it was really nice to see that it's working. And then a current project we have finished, we actually prepared some transparent gels, but also doped them with some cells, and we imaged them with our compound, so we're actually very happy about that. Well, as a conclusion to this part of the presentation, I could say that supramolecular chemistry, and particularly this host guest inclusion complexes, actually may give another chance to some compounds we previously thought that they are useless completely because of solubility or because of toxicity. So in the next part, I will not discuss the disadvantages of the fluorescent microscopy or advantages of the Raman imaging. I will just say that in our department we have a nice infrastructure which permits us to perform the experiments with Raman spectrometry, and we have a knowledge in assembling nanomaterials using the DNA or the nucleic bases. Well, you will ask, so we decided why not having these skills, why not try to assemble some probes which will be suitable for imaging cells by Raman spectrometry. Well, you will ask why DNA? Because, well, DNA is a really amazing material. You can do a lot of things with DNA. Synthetic DNA is readily available at companies. You can attach different molecules to DNA. You can design things with DNA. For example, you can design simple nanostructures by just putting up some tiles together, and then you will have them. You can design and assemble more complex DNA nanostructures, 2-dimensional, even 3-dimensional. Well, you can easily modify the surface of gold nanoparticle with DNA using the tile chemistry, and while these particles are extremely often used for assembling either some sensors or used as a drug delivery very often. Well, and last and not least, DNA can serve as a surfactant to disperse and also solubilize single-wall carbon nanotubes. And particularly this property is very important for the next of the presentation. Why? Because dispersed carbon nanotubes, they have a very nice and particular Raman spectrum with very particular bands in it. Well, if a carbon nanotube solution will be used for biomedical application, we can detect these bands and actually perform imaging, mapping of the surface of cells, for example, like some of the authors in this publication have done. But the only problem is to get this kind of beautiful images, you need very high concentrations of carbon nanotubes and very long time of scanning. But carbon nanotubes at a certain level became very toxic, so it's not suitable. Then there is a solution for that situation, is to use an enhancement of the Raman spectrum, and this is called the process of surface-enhanced Raman scattering. Its principle is that you position a molecule of interest, in our case a carbon nanotube, close to a metal surface, which upon radiation can create so-called hotspots, which exponentially increase this Raman signal. Well, that's what these authors have done. Actually, they proposed an interesting approach, a 5-step approach. When they finally succeeded to decorate carbon nanotubes with gold nanoparticles, they succeeded to increase the signal of the carbon nanotube and subsequently to image the HeLa cells. Well, we investigated all the available methods, we applied our knowledge, and we proposed a completely new mechanism of preparation of carbon nanotubes decorated with gold nanoparticles. Well, our approach consists of only 2 steps, and they're quite easy. The first step would be the functionalization of gold nanoparticles with a DNA strand. We just bought a strand consisting of 40 thymidines residues and a tile moiety at the end. And then we developed a protocol where we could obtain gold nanoparticles functionalized with DNA, but at a very high concentration. And when I say high concentration, I mean micromolar range concentration. This method has already an advantage that at this step you can choose the size of the gold nanoparticles you want to assemble afterwards. The next step, we just use these concentrated solutions of the gold nanoparticles decorated with DNA, mix it with the single wall carbon nanotube bundles, and they are bundles until they're sonicated, and then sonicate them together. During the sonication, the carbon nanotubes will disperse for a very short period, but the DNA overhangs will capture them, will stabilize them, and attract the gold nanoparticle towards the surface of the carbon nanotube, like it's presented over here. And the huge advantage of this proposed method is that not all the DNA from the gold nanoparticle is used for the stabilization. There are some sequences which are available for the post-synthetic labeling through hybridization to the complementary strand, which is very great because by this we can just introduce some targeted moieties, or we can introduce some tracing moieties for fluorescent molecules. So many things you can do post-synthetically with this solution of conjugates. Well, the design of experiment was done. We have monitored the performance of the attachment of gold to the carbon nanotube surface by just measuring Raman spectra of the reaction mixture every 30 minutes. At the beginning, we monitored two different bands from the carbon nanotube spectra, and we observed that at the beginning, prior to sonication, the carbon nanotube bundles were giving very broad and low intensity signal. After 30 minutes, the signal increased, and after 60 minutes, the reaction is probably getting to the saturation. Well, actually, I'm sorry. Actually, it was enough for 1 hour to sonicate and to obtain the conjugates. But we wanted to be sure that they are indeed formed, and for this purpose, some surface analysis is needed. And I'm talking about atomic force microscopy. We just deposited the solution on the microsurface, and we could see all over the surface this type of structures where we have tubes, and from place to place we had different densities of gold nanoparticles. What is important in this atomic force microscopy experiment is that we didn't see many of the gold nanoparticles lying around without carbon nanotubes. And this is very important actually. It means that all the used gold nanoparticles are decorated on the surface of the carbon nanotube. Next step was, of course, checking the toxicity of the obtained solution, the stock solution, and then 2 and 3 time dilution factor solutions were tested for cytotoxicity evaluation, and we observed that even starting solution was kind of okay, but with the dilution the viability of cells increased. And then we took the less concentrated solution and incubated it with HeLa cells, imaged them with bright field, choose a region, detected the G-band signal of the carbon nanotube, and then performed the mapping of the surface. Well, these are the images we obtained. I think they're beautiful. For us, it was very important that the signal was coming only from the surface of the cells. Well, we don't know and still don't know if the carbon nanotubes did penetrate inside the cell or they were stuck on the surface of the cell. We don't have the necessary infrastructure. But for us, it was really important that at this step we could see the Raman signal, we can map it, and we can visualize the immobilized cells. Well, so far so good. The next step was actually what I said, that we can post-synthetically attach targeting molecules or any kind of molecules through hybridization. And for this purpose, we just designed a sequence containing a fluorophore, which are complementary to any of these available overhangs of DNAs. And upon hybridization of this sequence to the surface of this conjugate, the dye should come in close proximity to the gold surface, and then it should be quenched. Not completely, but quenched. In a negative control experiment, we took a similar sequence, but which is not complementary to DNA. It means that it will not bind and it will not be quenched. Indeed, the analysis, we performed the fluorescence analysis, we performed all the corresponding two sequences prior to the addition of the conjugate, showed high intensity of fluorescence. In this case, the freshly prepared sample was added into the solution, the hybridization took place, and the signal quenched. In this case, since no hybridization was happening, it was just a small dilution factor decrease of the fluorescence. Again, so far so good, but then we sent the manuscript for the publication, and it was a question of the reviewer, an extremely nice question. He said, well, you say that you add the sequence and it hybridizes only to the free sequences of DNA. But what if this sequence also hybridizes to the sequences which stabilize the carbon nanotubes? So meaning that it will actually wrap the golden particle from the surface of the carbon nanotube, and you will have this something like this, and a bare carbon nanotube lying all over. Okay, nice, very nice question, very nice experiment. So what we did, we prepared a fresh solution, added a huge excess of the complementary strand, and then performed the AFM imaging. Well, nothing happened actually. We still could see all over the surface carbon nanotubes, and they were all decorated with golden particles as we were expecting. Nice, but we like this project very much. It was really interesting, but it has a very big disadvantage. We also stated in the paper, because as you can see, for example, from these two pictures, and if you compare them to previous pictures, you can see the difference in the density of the golden particles on the carbon nanotubes. And unfortunately, experimentally, we cannot control this density. It means that from sample to sample preparation, you will have different increasing of the signal, and which is actually not really good. So we put this project on hold for quite some time until we figure out a completely new material which will help us in our research in this area. And beginning of this year, actually, we paid our attention to a material which is found in the literature under several names. For example, like gold plasmonium black body, you can find out that it is colloidal gold. Black gold, you can just find as a black gold. And what are these? Well, it's very similar to what Professor Vekas just said in case of magnetic nanoparticles, where you have a cluster of the size between 50 and 150 nanometers, and this cluster consists of densely packed small nanoparticles. And they are so dense and so close that they absorb all the light from the visible region and infrared region. So if any of radiation comes on the surface of these clusters, it's immediately absorbed, it is scattered inside the network, and then eliminated in the form of heat. Well, sounds quite interesting because between them all the hotspots are formed. So actually we can perform interesting things with that. It looks like really black colloidal stable solution. They are stable for quite some time, very interesting. And they indeed, if you see, we performed several dilutions of the stock solution, but all the investigated solutions showed the complete absorbance of the visible region of the spectrum. The scanning electron microscopy also confirmed the formation of these clusters, and if you go closer, you can see that these clusters do have these small particles bind together in very dense form. Of course, the very logical next step was to perform the cell viability assay of the samples. Unfortunately, these are very raw data, they're not final yet. You could see some just markings over here, but they mean all the dilutions or different synthetic approaches to prepare it. But as a conclusion, they are non-toxic. You could see that their viability is quite high, so which is okay. Another interesting point is that these black materials can absorb different small molecules, and particularly the Raman probes, which are very easy. For example, the methylene blue. We just took a solution of methylene blue in a micromolar concentration, which in Raman spectra didn't show any of the peaks, absorbed it on the surface of these black bodies, and then imaged the Raman spectra, and you can see that the huge increasing of the signals coming from the methylene blue due to the multiple hotspots within these clusters. What we did next, of course, we incubated the cells with the clusters with absorbed methylene blue on it, and selected an area, imaged it with Raman, detected the signal, and obtained something like this. Well, it's completely different from what we saw in the carbon nanotube experiment. So now we see some accumulations, we see some different concentration, which is very attractive and interesting. But the specialist who is doing the Raman spectrometry in our group, she said that something is wrong. Something is very, very wrong, because the signal is not stable during the imaging. the signal is moving from part to part. Different other signals have appeared in the imaging. And we said, like, okay, let's check what happens with the sample. And then we just imaged again with a bright field, the same area, and we could see that the dead cell was completely damaged. We could see that just all over the places where actually we saw the signal was extremely damaged. Well, since, as I mentioned, these compounds absorb light and transform it into heat, we immediately suspected that localized irradiation created localized heat, which completely destroyed the places. We confirmed this theory by an experiment. We took completely random 2 cells incubated with this black body. We irradiated them for only 10 seconds in only one single place, and you can see that depending, of course, on the quantity of this black gold absorbed inside the cell, you could see anyway the damage of the cell. So now, as this project is ongoing, we have a lot of open questions. Among them, one of the questions is, maybe we are choosing the wrong application. Maybe we should go better to the area where we can use it as photothermal therapy instead of doing imagistic on it. But we still hope that maybe we can adjust the experimental conditions in such a way that we can first still image the cells and afterwards kill them. That would be really great to be a theranostic application of the system. Sounds quite interesting, but it's still an open question. And with this slide, I would like to finish my presentation, but of course not before mentioning some great people who were producing the results for this presentation. They are both PhD students, junior researchers, and experienced researchers, collaboration from neighboring university, collaboration from abroad, and of course the money, which will come from the national project and from European projects also. And all this money came in several years, so we can obtain the results continuously. And with this slide, I would like to thank you for your attention.
Thank you. Thank you. We have time for 2 very quick questions. Yeah.
Nice talk regarding the fluorescent compound based on cyclodextrin.
Yes.
So I see the mechanism of this dye going in the cell. Do you have any mechanism which stops the dye coming out? Did you observe any linkage?
No. After 24 hours of incubation, we observed this formation of bright images, and they were stable for 72 hours. So it was really stable complex. We don't know the mechanism indeed. We don't have the infrastructure to investigate it, but actually it was really stable in time. What it is what we saw.
Another question? Comment? Yeah.
Did you try to see how temperature difference hybridization of gold nanoparticles fixed to carbon nanotubes and single-strand DNA added after?
No.
Hybridization. How temperature?
Since it was quite a long sequence of 40 bases, the melting point of DNA typically should be quite high, above 50 degrees, I think. Because, well, there is a temperature calculator to check it, but we didn't check it because typically sequences of 24, 25 to 30 are melted down at 40 degrees. So it means that 40 bases long DNA, it should melt quite high. So, but no, we didn't measure that.
Okay, thank you again.
Thank you very much.
The next presentation comes to
change, but I have to put my computer. No, you cannot, because I have a movie.
Professor Sigan,
I'm not sure what is it.
Faculty of Physics.
No, am un filmuleț și ăsta nu știu dacă poate demara de pe alt calculator și le-am înregistrat. Bun. Da, de asta zic că se conectează, nu știu Da. Not now.
Preliminary informația de
Thank you. Introducing me. I'm really happy to be here again. Just I say again because last time when I was here was 8 or 10 years before, I suppose, in the almost same community. Thank you, Eugene. Thank you for the organizer for giving again this possibility to update my research. I remember I started also with last morning, but since then I add more results, and I will just introduce a little bit my research group. I'm in a certain specific point of my career. I am grateful to these nice people here because they are my former PhD students, current collaborators, almost 25 PhD students since now, and I will not present here the most recent results, but I think I pick up the most significant almost from each publication just to give a flavor of research in my group. This research was done also in collaboration with a group in the Institute of the Interdisciplinary Research on Bio-Nanoscience. And then here is Professor Popescu. His group collaborates currently with my group. You have several publication results, and so Professor Simon was here again, his group collaborated. I have also collaboration because it's almost in bio application with a biologist and chemist from my university, also from Institute, Oncologic Institute in Cluj-Napoca. So now my research since 15 years it was focused on using some kind of nanoparticles. Today they are called plasmonic nanoparticles, but I mean 20 years or 15 years before they are colloids. Now the name is more in plasmonic, nanoparticle, or so on. They are very useful, in fact, because they offer several optical properties very rich in application in biomedical field. So starting, I will just present a short introduction of what are this kind of surface plasma resonance. So then I will present a flavor of our particle, a nanoparticle, which I prepared in my laboratory, and I selected a specific application in sensing, cell imaging, and some possibility for therapy by light activated therapy. But what about surface plasmon resonance? This is a basic text explanation. When light interacts with a free electron in the nanoparticle, they created in terms of classical physics, a dipole, and then, but the optical signature of this interaction, there are several optical signatures of this interaction. One is increase the local field around the nanoparticle. So the local electromagnetic field around the nanoparticle is increasing. And by, let's say, 10 power 3, it's usually too big. But this is not only. Again, there is some kind of light which is absorbed by the particle. The signature is by a spectral absorption, by spectrum. Here, I think I can use that. Here you can have a which is called surface plasmon resonance, localized surface plasmon resonance for round shape, golden nanoparticle, standard particle. So it's very good for sensing. If something is happening around the nanoparticle, it will be shifted. Again, this spectrum is very dependent of the size and the shape of the nanoparticle. So you can move, you can tune the resonance from almost UV to near infrared, 1000 nanometers. And third application, there is no light absorbed, but light as a dipole is like a small antenna, scatter light. Then you can see this nanoparticle when this is a movie. I think this work. Look inside the cell. You can notice the Brownian movement of this nanoparticle, and also the color change. The color change means the interaction with the molecule in the cytosol, or even this is a dark field microscopy, and scatter light by the cellular skeleton overlap with the scattering light from the nanoparticle. Maybe, yeah. Now, this is intrinsic signal from the particle, but again, now another interesting signal is luminescence. The gold nanoparticle was unknown last 10 years before. They are also fluorescent, not fluorescence, the term is luminescence. They are luminescence, but are luminescence when are excited by 2 photons. In pulse laser, 800 nanometer by 2 photons, by recombination, transfer between SP band to D band of the metal, they give light. So you can notice here also visualization of the gold nanoparticle captured inside the cell without any labeling. Only nanoparticle itself and scanning with our microscope, time resolve microscope, you can notice the particles. Now, other applications are connected with the fact that the surface plasmon excitation can improve the detection of other molecular or entities in connection in vicinity of nanoparticles. Already my colleague presented this effect, which is very, very useful. This is called surface enhanced Raman spectroscopy, and in between the nanoparticle is a specific hotspot that the field is here even higher, And so by this relation you can understand the intensity is enhanced, and the optical signature, spectral signature is also enhanced. Also you can detect even single molecule, single molecule in this kind of hot spot. The second effect, which is also surprising, it's known that the molecule touch the metal will quench the fluorescence. But in fact, if the molecule is at certain distance, a space of few nanometers far from the surface of metal, you can couple the molecular dipole of the nanoparticle with metallic dipole, and then in this coupling there is a energy change. You can improve the emission of the molecule. You can modify the rate of emission and gain more light. Even a molecule with a quantum efficiency very, very low, they become very bright in the vicinity of nanoparticle. This is similar like cells, but a little bit there is some kind of difference in definition. Now, this is about effect on the nanoparticle, but then from there you can have several interesting applications, what is called today in nanomedicine. Using in therapy and diagnostic, or even the new word, diagnostic, which can be composed from therapy and diagnostic. You can do hyperthermia because, as I said, the nanoparticles are so light and heat up the nanoparticle is heated up. Also, they can be used as vehicle to delivering drug inside the cell, and coupling plasmon with a specific molecule called photosensitizer can improve the photodynamic therapy of photosensitizer, and so on. There are several here. But for having this kind of particle applied in the biomedical field, there are several requirements to do after synthesis, in solution. They should be stabilized and biocompatibilized by polymer, coated by the specific polymer, biopolymer, and then so they have to be targeted for a specific molecular receptor. Again, they can be tracked inside the particle by using some reporter molecules, very small molecule by cells. This is one example. In our interest, in my research group, we try to control the resonance of nanoparticle to bring this resonance inside this window called biological window, because light in near infrared, since 700, 900, 1000 nanometer is more transparent, and the background from the tissue, if you think to apply in real life, then it's better to work with nanoparticles which have resonance in this window. This is our interest. Now, this is about the introduction. Now, what kind of nanoparticle and call later on nano platform will you fabricate in my laboratory? I remember at the beginning, it was the first time when I arrived here, I discussed about this kind of solid substrate. I will not speak today about solid substrate, but they are also useful in cells as substrate, solid substrate. You are more interested now, and my presentation is focused on this kind of nanoparticle, which are prepared in solution, usually in water solution, to be easily transferred to cell. They look like them depending on the color. Color is linked with the shape of this nanoparticle, and stabilization is also a very important point. My colleague previously discussed about this aspect. Is also important for our particle to be also stable. Sorry, just I have to skip this presentation because I forgot to hide this presentation. It belongs to another kind of presentation. So just now I will speak about plasmonic nanoparticle in solution because they are convenient for biological application, not on substrate. Let's see some of them. Gold nanorod. Gold nanorod are anisotrope in terms of their length relative to this, for the size, their size. In this case, because you have two dimension, you have also two local resonance. One is a transversal, located more around 500 nanometers as a round shape, and the second one resonance is longitudinal on the particle. So when you use unpolarized light, you will get every time this kind of double resonance, which can be controlled very nice by the length of nanoparticle, and the ratio aspect show very clear. So you can cover optical domain here invisible. Each of them will be applied later on, show some application, but now another kind of particle is when have tips, but they are in fact pyramids. Because there are two big pyramids, they are called big pyramids, nano bipyramids, like rice, look like, but in volume. And then here are also the very important is also the size of this and the width of the band, so you can control also the position, but also the width of the band. Again, on the overlook far in infrared. Each one have a specific application. Now, a hollow gold silver nanosphere are very interesting because you can put inside some medical some drugs and delivering these drugs. They are empty inside at the beginning. Now, this kind of nanoparticles are made of silver and gold, a core shell. I mean, shell is silver in this case, and core is gold. They are broad, are individual nanoparticles, but they are very large resonance band here, which is also convenient to excite if I do some kind of hyperthermia. This nanoparticle was done on gold. Now in silver, I also worked several times in doing some kind of nanoparticle in silver triangular shape. You can see, look at how the shape of the nanoparticle, the same material silver, but the shape change from round to prismatic triangular shape, and the spectrum also shift from 400 to 800, and several here notice for this kind of triangular nanoparticle, there are several resonances. The symmetry is different, so you can have 4 specific resonances. Now, this is almost all I mean about the kind of nanoparticle you prepare currently, you can prepare in our laboratory. Now, some application of these nanoparticles. So is sensing by cells and doing also imaging. Now, to avoid heating up the nanoparticle, my colleague speak about the fact that there is aggregate and the black body absorb light and heat up and destroys the cell. So the solution is to use individual nanoparticles, which in fact has corner, tips, whereas the cells is better amplified. So this is the reason that you work for doing cells not by aggregation, but by individual nanoparticles. This is, first of all, here you prove an example. The nanoparticle are inside the nanoparticle inside the cell by dark field microscopy. And now this is the design of our nanoparticle. So we use triangular because there are several resonances protected in the chitosan, coated with chitosan, and also attached to the metal is a small molecule, aminothiophenol, which connect by thiol with the metal. So this kind of nanoparticle inside the cell will give the signal, the specific signal from para-aminothiophenol with a very few bands and can be easily understand. Here is not very clear, but it's a bright field imaging of the microscope, bright field microscopy. And then here, and even here is Raman map of the same cell, which is here, single cell imaging. We can identify by specific band not only the presence of nanoparticle, which is marked in red, here in red, inside the cytosol, but also you can detect the cellular component, nucleo, mitochondria blue, and it can map entire the cell, and specifically find the nanoparticle. This is the no heating, and you can repeat this kind of experiment. Now I will select some application regarding using light-activated therapy. The first one is by hyperthermia. First, you have to prove. Then, using here, in this case, you notice these individual nanoparticles. These are not cluster, individual nanoparticle, deep pyramids, nano deep pyramids, which express a very strong, narrow band in near infrared, specifically at 755, where you have the diode laser, and prove, first of all, you prove in solution, then by heating with laser this solution, and by camera, infrared camera, you can follow the increase of temperature inside the vial there, and so sure we can explain the increase of temperature by thermodynamic equation, laser absorption, and sure by specific parameters of the metal, and you can simulate this spectrum, also compare with the experimental results. What is the conclusion here? Then you can increase the temperature only by maximum 20 degrees or even 10-20 degrees, but this is enough starting from physiological temperature of 37, so with 10 degrees it's enough to induce apoptosis in the cells. And you can repeat this experiment. Now this is demonstration in solution, and you did this for several type of nanoparticles, all these particles were in near infrared, at about 800 nanometer, was a triangular shape are here. This is cell mortality in function of laser intensity, and triangular nanoparticle and nano-rod nanoparticles. So the most efficient by optical properties are silver nanoparticle. Sure, the demonstration is done by staining the dead cell relative to experiment. There were several experiments for comparison and to have this result. Sorry. Now, the previous experiment was done without any targeting. I mean, the nanoparticle was covered by chitosan, and but no target, no specific molecule to target. In a second demonstration, you did specifically perform targeted photothermal therapy. For them, there are, you work with cancer, ovarian cancer cell, which express receptors and folic acid is conjugated by folic acid. You address specifically nanoparticle, the same nanoparticle, triangular nanoparticle, a reporter for paraminothiophenol, then folic acid to recognize the receptor expressed by the surface of the cell, and then sure you start to do this kind of targeted therapy in terms of cell study, level of cell, using the laser 800 laser, and then what was also possible to do simultaneously also measurement not only for inducing hyperthermia but also for localize the nanoparticle inside by meaning of cells spectrum. And here also some dark field experiment where you can prove then by conjugation with folic acid there is a much more density of nanoparticle localized on the membrane of the cell relative to non-conjugated with acid folic. And also the specific band help us to induce and to recognize the position in the cell. Now this was a single therapy, hyperthermy, targeted and not targeted. Another interesting result you developed in our group was to do multi-therapy. I mean, using a combination between what have you only done hyperthermia, heating up the nanoparticle, but having in the polymer also a kind of molecule which is known in literature photosensitizer, which generate reactive oxygen species. Then having together this effect, you can do some synergy in treatment by combination hyperthermia with PDT, and because it's more efficient, you can lower the amount of photosensitizer, which are side effects, in fact. And so this was the first motivation. And you discover there are some interesting effects unknown before, I will say, the presence of photosensitizer in the local field of the nanoparticle reduce what is called photobleaching. Photobleaching can be reduced just because lifetime on the excited state is shorter, and then you can improve also the transfer to triplet state. And then finally enhancing singlet oxygen generation. Also, when you use with a cell, polymer here is a kind of pluronic, one pluronic block polymer, which is useful in this kind of studies. Here you prove also by comparison control sample, live cell, not irradiated with our nanoparticle, and here you can use with nanoparticles, so you can see effect between here by staining the dead cells. Also reducing the intensity, you can see reduce rate of mortality. Now, this study before was done with a single particle, and you notice, in fact, that it will be possible to use small but very small aggregate, not huge aggregate, small aggregate, as their size is under 100 nanometers, made of 20 about nanoparticles, which can capture inside and create hot spots to also promote the possibility to map by cells. And notice also interesting effect, some of the methylene blue, in this case, which is a photosensitizer you check, is not located on the hot spot. There are some molecules which are linked on the polymer, and they, being placed in the local field, they can be amplified by metal enhanced fluorescence mechanism. Also, there are possibilities to prove their presence. Look here, individual nanoparticles in red, then a small cluster of nanoparticles a little bit shifted to 600, then having here the presence of methylene blue cell spectrum, also the emission, the fluorescence emission. Then you can characterize better in solution. This was done first of all in solution, then was tested and checked on the cells, and here just to prove dark field microscopy for cell with our nanoparticle and incubated for 3 hours, I mean, and maybe 6 hours, I don't remember exactly now how long was here. Here in the this case we can have the nanoparticle. Now, using this kind of small nanoparticle, small cluster of nanoparticle, you are mapped to do what is mean tomography. Tomography in the cell. I mean Raman tomography, which means then you scan the cell. You can scan the cell with a bright field image under Raman microscope at different Z position, and then using and treating the band, methylene blue band, you can notice from up to 0 from 8 micron, almost in 2 steps, the position, how the position of nanoparticles are inside. And sure, here there is a little bit more an increase of efficiency. I'm sorry, I cannot see here on computer. I can notice here the red dots, but here I don't see, can't see. But the study was done also by comparison, not treated in irradiation and treated only to gold nanoparticle and only to methylene, and together, and you can notice the synergistic effect in destroying the cells. Another study would, in another kind of study, in another kind of nanoparticle, use bipyramid, nano-bipyramid, where you do also PDT, I mean photothermal therapy, photodynamic therapy in melanoma cells. Again, here you can prove the area with a dead cell and the live cell, just separate here, whereas the laser is the border of the laser on the substrate. Yeah, now you use a nanoparticle also as a vehicle to deliver a drug. One of our study was concerned using delivery for doxorubicin, which is a chemotherapeutic drug. And the procedure was to capture this doxorubicin in the polymer shell in the nanoparticle, and then prove the this is a gelatin around, the shell was a gelatin. You can notice here in Thalian microscope, in electron microscopy, the size of nanoparticle, and greenish here is a gelatin. The spectrum show also that the advantage of this kind of drug is that the doxorubicin is also fluorescent. So has itself a signal. Yeah, you can notice then when you measure in the same concentration spectrum, fluorescent spectrum from the free doxorubicin and the doxorubicin capture on the shell, on the gelatin shell. There is some decrease in intrinsicity, which is usually. But what is more interesting, you done a study of delivery, because it's not enough only to charge, to load the nanoparticle with the drug, but it's very important to delivery. And to control delivery, we modify the pH of the solution. This is known. Then in cancer environment cells, the pH is more acidic. So it is true here. Then at neutral pH, there is a degree of delivery, but increase significantly for some pH around 5.
5 minutes, please.
Yeah, just maybe use 4 minutes. So, and then also by increasing the temperature. Temperature under physiological temperature we can lose, but also increasing up to 35, there is also delivery or release of doxorubicin. So this was controlled by intensity of fluorescence. Yeah. Here is an example of using multimodal imaging of the same nanoparticle. So using dark field, this is very clear by dark field. Here you can measure scattering from the nanoparticle, because our microscope can also capture the spectrum from the nanoparticle. When you have some emitter like doxorubicin of quantum dots, also you can measure, you can image by fluorescence lifetime imaging. So you can localize doxorubicin after delivery, doxorubicin go inside the nucleus. That's very clear. Then here is the position by green, it's doxorubicin inside the nucleus. Then also sure by Raman also can map the position. So this is using the same on multimodal imaging, doing spectroscopy, microscopy in confocal system. Our confocal system are convenient. Another example, we're using carboplatin, which is also a chemotherapeutic drug. It's individual silver nanoparticle, reporter molecule. Here you use mercaptobenzoic acid as a reporter molecule, which is very sensitive. Mercaptobenzoic acid is very sensitive to pH solution. So if in the side of the cell, here you can notice the localization by red spot. Red spots are assigned to the nanoparticle, and the blue and yellow are nucleus and membrane and so on. This is imaging cells, images of the cell. But very interesting results here when you look specifically at the band of mercaptobenzoic acid. You can notice where is a signal from more acidic or neutral inside the cell. So you can map pH inside the cell by using a specific reporter, benzoic acid, mercaptobenzoic acid. Yeah, so I mean, now this was done almost, I mean, last 10 years where I speak. Now, in my research group, we are developing now a new project. It's almost at the end, but when I decided to come here, I decided to show again what we did more from the previous my presentation with a focus on plasmonic. That was the reason that I presented here only on plasmonic results. But now you have also several new results on using some specific contrast agents to map to do real time image in surgery, and specifically in ovarian cancer. So this may be another occasion, but there are some publications in literature about progress on this field, which is at the end of our final project in September will be covered. Now, as the finish here, as you notice, you implement several experimental and simple methods to prepare a nanoparticle in which you integrate some functionality in sensing, drug delivery, therapy, and the current research work and future work is to translate more in vivo, in vitro, in vivo, and sure for doing some intraoperative plasmonic guidance. We are at the end, and thank you for your attention. Okay, so I have. Thank you.
No questions?
Okay.
Okay.
Thank you.
We have a coffee break, yeah?
No, one question.
microorganisms and tumor cells. So we are moving a little bit the profile of the speakers towards the end users, let's say, of these wonderful methods that you have presented us, and we are very grateful to you that you have found the way to present these very complicated methods in a very accessible way for us. Thank you so much. And now we are coming towards you and we present you the challenges that we meet in the biomedical field. So we will start with the first presentation made by Dr. Florescu together with Dr. Nika about the phenotypic and molecular methods used today in bacterial identification and antibiotic resistance testing. Please, you have the floor.
Thank you. So I have the pleasure to attend this beautiful conference today, and thank you for your invitation. And I will switch a little bit the way of looking into things from the initial point of view, because I'm an infectious disease clinician, and I made this paper with this presentation together with my colleague, Dr. Nika. She is the head of the laboratory in the Victor Lovesh Hospital, and she is also a microbiologist, and a very good one. So we are going to talk about bacterial identification and antibiotic resistance techniques used today in our laboratory, and the one that we intend to use if we will be capable of from other points of view. So, which is the role of the microbiology laboratory in the infectious disease hospital? Well, it's a very important role, and I think it's a central role, it's a very main role, because in an infectious disease hospital you have to have a diagnosis. It's the principal movement for the treatment of the patients, the diagnosis, and the bacterial diagnosis also. And the principles for a clinical microbiology laboratory is safety and efficiency. These are the main principles, and also the flexible design, so you can adapt very quickly and from time to time when you need this to adapt the specific of the laboratory, the circuits, and so on. And as we all know, the microbiology undergoes very rapid changes, and the clinical and epidemiological consequences are evident. And as we all know, the problem of emergence and re-emergence of various infectious diseases, the one, for example, which we are getting through now, or other type of bacterial infections, emerging resistance, it's a very important problem, and it's an international problem for which all the states are doing all the possible measures to keep it very little as possible. As we know, for biochemistry and hematology, we already have a lot of automatic processes in the labs. Everything is getting very automatic and more smaller as physical dimensions and so on. But why the bacteriology laboratory is very slow on automation? There are some reasons. The variability and complexity of diagnostic samples. So in bacteriology, you have to deal with samples from all kinds, liquid, solid, different types of tubes, of transport media, and so on. The variability of biological sample transport containers, they are also important because if you want to have a good diagnosis, bacteriological diagnosis, microbiological diagnosis, you have to transport the samples correctly to the laboratory. Because if you don't respect this stage of diagnosis, you will have a false negative diagnosis also. And not the least, the large number of bacterial pathogens are involved for this kind of diagnosis, and this requires all kind of techniques, laboratory techniques. But I think the main role is the microbiologist, because with you have to have a microbiologist for diagnostic interpretation. It's not very easy to make a very automatic bacteriology laboratory, because all the final results must be interpreted by a doctor, a specialist, and to tell to us, the clinicians, the role of the etiological factor that we find or should find. Is it a pathogen? Is it a positional pathogen? Is it a colonization? Is it an infection? And this cannot be automated only in few cases. So, will automated microbiological diagnosis replace conventional diagnosis? Maybe, but I think that not all the diagnoses. We have the classical bacteriological diagnosis, which has obvious advantages. So it's very high sensitivity, safe, low prices, and has also qualitative and quantitative results, which helps us to differentiate the bacterial populations, the pathogenic, the commensal flora, and so on, from the mixed flora existing in a pathological sample. And you can have the antibiogram, you can quantificate the resistance of the pathogen, which is very important nowadays. But it also has some disadvantages. It's very time consuming, so it says 1 until 3 days, but it can take more than 3 days in most of the time. And it needs successive stages for the isolation of the culture of diagnostic interest. For example, you all know that for tuberculosis, for TB, the classical type of microbiological culture needs 60 to 90 days, so it's a very long time. And is sometimes limited by the number of viable microorganisms in the clinical specimen. Is it well, the specimen is well taken or not? The antibiotic treatment prior to preservation, because if you treat a patient with antibiotics for an infection, and after that, after 1 or 2 days, you are going to take a sample to make a microbiological diagnosis, you will not find anything. It's a false negative result, and you cannot identify the etiological factor, and you have to go with a maximum antibiotic scheme until the final treatment. And the microorganisms with intracellular grow, or they do not grow on artificial media in vitro, are another disadvantage for these classical cultures. You cannot grow this type of microorganisms in the classical cultures. So, what about automatic microorganism detection systems and biological sample? We have that. It's a very rapid and sensitive method. For example, for the detection of bacteria and fungemia, the clinical role, the essential role of the clinical microbiology laboratory, it's obvious, because we have to have a blood cultures result. I think the main disease in the infectious disease hospital, the sepsis and very complicated other types of infection which are going with bacteremia and fungemia. We saw that now in the pandemic of COVID. And we have to have a rapid answer, because we have to make a very targeted antibiotic treatment for these patients. So we need this also for guidelines or global guidelines or national guidelines, and also for surgical procedures. The surgeons know better that they need this type of, for example, cardiac surgeons, for patients with endocarditis. We have to have a result for them and to tell them how can we go with the antibiotic treatment eventually together with the surgery. But not always a blood culture is has also the etiological pathogen like that, because it requires correlation. So if we have a positive blood culture, it doesn't mean that this is the disease of the patient. We have to correlate it with the clinical data, with the imaging data, with the inflammatory or infectious markers to see if it is really an infection, if it is this, the etiological pathogen that we are trying to find, or if it is only a coincidence or it is a false positive result. So all this data we have to correlate with the clinical part. That's why in our hospital, the clinicians are always talking to the laboratory doctors, bacteriologists mainly, to find out all these correlations. And also, there are cultural methods or direct diagnosis, isolation of bacterial or fungal cultures directly from the samples. We have in the hospital the automatic blood culture systems. We have BactAlert. There are also other methods like BacTech and VersaTrack, which each of them are methods that are detecting the bacterial growth in the blood culture from diverse kind of technique, colorimetric method, detects oxygen consumption, or identifies change in pressure and gas composition resulting from microbial metabolism. So we are incubating these are the BacTech, BactAlert used in our hospital. We are incubating the blood cultures in that device, and that device shows us if it is positive or negative. And after that, the laboratory doctors are doing the identification of the microorganism. We can do also the molecular diagnosis, bacteriological diagnosis. This means the partial or complete detection and characterization of DNA or RNA in clinical specimens. This is a very known technique nowadays, and a lot of hospitals are doing it. It is useful in microbiological diagnosis, but also in surveillance, epidemiological surveillance, in research, of course. And the advantages are the identification of uncultivable or completely unknown pathogens, because we are having more and more situations like this in our hospital, when the Dr. Nika is calling us and telling us from a variant of Staphylococcus, for example, which we never heard about, and it's a very new variant and is detected by this kind of techniques. And also is reducing the dependence on cultivation techniques. So it's a superior method, very fast, very reliable. Advantages of the molecular techniques is standardization, accuracy, automatic rapidity, and this makes it an extremely important part of microbiological diagnosis in routine practice, because we are using it in routine practice nowadays. Examples of molecular techniques in microbiological diagnosis, some of them, of course not all of them. For example, the diagnosis of infectious disease with slow-growing microorganisms like Mycobacterium, which, if you remember, in classical countries we have to wait 2 or 3 months for the diagnosis. This is made in 1 to 2 hours. Chlamydia, Bordetella, diagnosis of infections with non-cultivable bacteria, which we cannot get in the blood cultures, like Rickettsia, Trophilima, and other types of non-cultivable bacterial species. Another example is identification of toxic producing species or detection directly of toxins produced by, for example, a very present disease in nowadays, Clostridium difficile. The colitis with Clostridium difficile is a main problem of the nosocomial hospital-acquired infections, like Staphylococcus aureus and other types of Enterobacterium C. The screening of multi-drug-resistant bacteria is made through the molecular techniques. The diagnosis of partially tick infections, if you remember, I told you that in the classical infection, if we treat the patient with 1, 2, or 3 doses of antibiotics prior to the sample, to send the sample to the lab, we will get a negative result. But in these cases, we can do this, and we still have a positive result, but we will not have the antibiotics. And the detection of antibiotic resistance genes directly from the clinical sample is another very important possibility to use this kind of techniques. The macrobiological molecular diagnosis is based on the detection of nucleic acids, and we have some classification of the main methods here. Used methods without amplification, target amplification methods, nucleic acid sequencing, and automatic microorganism detection system in biological samples. And if we talk about first one, the methods without amplification, I try to keep it very simple, definition and all the data, because the first version of this presentation had around 100 slides, so it was not possible to talk that much. So this method without amplification means that the consists in hybridization methods with oligonucleotide groups. And for example, the DNA fish technique, DNA fluorescent in situ hybridization, in which the samples are marked with fluorescent dye, which can be viewed under